Welcome to the deep dive. Today we're getting right into the guts of it, literally. We're looking at the gastrointestinal system.
Yeah, the GI tract. And we're using chapter 36 of Porth's Essentials of Pathophysiology as our guide here.
Foundational stuff.
Absolutely. And our goal today is, well, to see the GI tract for what it really is. Digestion, absorption, sure, everyone knows that.
But there's more.
Oh, much more. It's the body's biggest endocrine organ. Think about that. Hormones galore. And it's also this massive immune barrier. Huge surface area to defend.
That's a key point, the barrier idea. You have to kind of reframe how you think about it. It's basically a tube running through you, right?
Exactly. It's technically outside your internal environment, the food, the microbes, all that stuff in the lumen. It's not in you until it's absorbed.
It's the ultimate gatekeeper system.
Perfectly put. And this tube, this specialized pathway, it's organized. You've got the upper part, mouth, esophagus, stomach. That's for intake, starting the process.
In the middle section, the real workhorse. Duodenum, jejunum, ileum.
Right. That's where most of the heavy lifting for digestion and, crucially, absorption happens, like 90% of it.
And then you wrap things up with the lower segment.
Colon, rectum mainly.
Right.
Water absorption, storing waste, getting rid of it. But, to really get how it all works, you have to start with the wall itself, the structure. Those 4 layers are everything.
Okay, let's peel back those layers. Starting from the lumen, the inside moving out. First up is the mucosa.
The interface layer.
Yeah.
It's doing a lot, secreting mucus for protection, making enzymes, absorbing nutrients, and it's got lymphoid tissue for that first line of immune defense.
And you mentioned something about its speed, how fast it turns over.
Oh, it's astonishing. The epithelial cells lining it, they regenerate completely about every 5 days.
Wait, 5 days? That seems incredibly fast. Why? What's the point of that kind of turnover?
Well, think about what that layer faces. Constant abrasion from food, harsh stomach acid, digestive enzymes trying to break things down, trillions of bacteria. It's a war zone.
Okay, yeah.
So this rapid turnover means it can repair damage incredibly quickly. If there's an injury, like from a toxin or maybe chemotherapy, which hits rapidly dividing cells, it can bounce back fast once the insult stops. The speed is the defense, in a way.
That makes a lot of sense. Okay, so past the super fast regenerating mucosa, we hit layer 2, the submucosa.
Right. Think of this as the support structure. It's connective tissue, but it's packed with the important stuff. Blood vessels bringing nutrients and taking away absorbed products, nerve fibers, and glands that secrete enzymes and fluids. Sort of the utility layer.
Got it. Then layer 3.
The muscularis externa. This is your movement layer. It typically has 2 layers of smooth muscle, an inner circular layer and an outer longitudinal layer.
Circular and longitudinal.
Yeah, working together. The circular layer constricts the tube, pinches it. The longitudinal layer shortens it. Together, they create those mixing and propulsive movements, peristalsis.
And finally, the outermost wrap.
The serosa. Or, in the abdomen, it's continuous with the peritoneum. This is that big, serous membrane lining the abdominal cavity.
And connected to that, or part of it, is this thing called the greater omentum.
Ah, yes, the greater omentum. It's pretty unique. It's this large apron-like fold of peritoneum, hangs down from the stomach. It's fatty, stores quite a bit of fat, actually.
But it does more than store fat.
Oh, yeah. Its nickname is sometimes the policeman of the abdomen because it's mobile. If there's inflammation somewhere, like appendicitis or a perforated ulcer, the omentum can actually move towards that spot.
It moves.
Yeah, it migrates and tries to stick to the inflamed area, forming adhesions. It basically tries to wall off the infection to contain it and stop it from spreading throughout the whole peritoneal cavity. It's a fascinating protective mechanism.
Wow. So protection built right in.
Mm-hmm.
But the main job is movement, getting stuff through. How does this tube actually control that movement? It's not just one kind of squeeze, is it?
No, definitely not. You generally see 2 main patterns. There are rhythmic movements. These are intermittent contractions, like waves. You see them in the esophagus, parts of the stomach, the small intestine. They mix the food and propel it forward.
Okay, rhythmic. And the other type.
Tonic movements. These are different. They're characterized by a constant level of contraction or tone. They don't relax completely. You find these mainly in the sphincters.
Like the lower esophageal sphincter, the valve between the esophagus and stomach.
Exactly that one. Or the pyloric sphincter, or the internal anal sphincter. Their job is to maintain pressure, act as gates, control passage from one section to the next.
So how does the muscle know when to contract rhythmically or tonically? What's driving it?
Well, the GI smooth muscle is pretty special. It's mostly unitary smooth muscle, meaning the cells are electrically connected through gap junctions. They act like one unit. And deep within the muscle layers, there are specialized pacemaker cells.
Like the heart has pacemakers.
Sort of, yeah, but for the gut. These cells generate spontaneous electrical rhythms called slow waves. They're not quite action potentials, more like oscillating waves of membrane potential.
Slow waves. And these dictate the rhythm.
They set the maximum rhythm. The slow waves determine how frequently that segment of the gut can contract. But whether it actually does contract strongly depends on other signals, like nerves and hormones.
And the rhythm changes?
Oh, yes. The stomach, for instance, has a slow wave frequency of about 3 per minute. Relatively slow for churning. But get down to the duodenum, the first part of the small intestine. It ramps up to maybe 12 per minute. Much faster for mixing and moving things along quickly there.
That's quite a difference. So what provides those other signals, the go signals, on top of the slow waves?
That's where the nervous system comes in. And the gut has its own nervous system embedded right in the wall. The enteric nervous system, the ENS.
The gut brain we mentioned earlier.
That's the one. It's remarkable. It's complex enough to manage most gut functions locally, almost like a separate brain just for digestion. It has 2 main networks, or plexuses.
Right, the plexuses.
You've got the myenteric plexus, also called Auerbach's plexus. It sits between those circular and longitudinal muscle layers. Its main job seems to be controlling motility, the muscle contractions along the length of the gut.
Okay, myenteric for movement. What's the other one?
The submucosal plexus, or Meissner's plexus. This one's located in that submucosal layer, the support layer. It seems more involved in controlling local conditions, things like secretion from glands, absorption across the mucosa, and even local muscle contractions within a segment.
So the ENS is the local boss. But does the main brain, the central nervous system, have any say?
Absolutely. The ENS runs the day-to-day, but it's heavily modulated by the autonomic nervous system, the ANS. Both branches play a role.
Parasympathetic and sympathetic.
Correct. The parasympathetic system, mostly traveling via the vagus nerve for the upper and middle gut, is generally excitatory. It tends to increase motility, ramp up secretions, relax sphincters. Think rest and digest.
Makes sense. So the sympathetic must be the opposite.
Largely, yes. The sympathetic system is generally inhibitory to gut function. It decreases motility and secretions. But importantly, it tends to increase the contraction of those tonic sphincters, tightening the gates. Think fight or flight digestion isn't the priority then.
Okay, so you have the intrinsic rhythm from slow waves, the local control from the ENS, and then the override or modulation from the ANS.
That's a good summary. And you see this interplay clearly in things like swallowing, which has voluntary and involuntary functions, or in gastric emptying.
Gastric emptying, controlling how fast the stomach lets food out.
Exactly. The stomach mixes food with acid and enzymes, turning it into this semi-liquid stuff called chyme. Then the pyloric sphincter, that tonic muscle at the stomach outlet, carefully controls how much chyme squirts into the duodenum and how quickly.
And when that control goes wrong,
Problems.
big problems. For example, if emptying is way too fast, especially after some types of stomach surgery, you can get dumping syndrome. Hyperosmolar chyme hits the small intestine too quickly, pulling massive amounts of fluid in, causing cramps, diarrhea, dizziness. Nasty. And the opposite. Too slow.
That happens too. Gastric atony or delayed gastric emptying. You see it sometimes in long-term diabetes where nerve damage affects the ENS or ANS control. Food just sits in the stomach, leading to nausea, vomiting, feeling full really quickly. It's all about that carefully regulated timing.
Which brings us back to control signals. It's not just nerves, right? You mentioned hormones earlier. The endocrine function.
Yes, hugely important. Remember, the GI tract is the body's largest endocrine organ. It uses hormones released into the bloodstream, true endocrine signaling, but also local chemical signals that act on nearby cells, which is called paracrine signaling.
Okay, let's talk key players. Which hormones are running the show? Starting with hunger.
Good place to start. That's primarily ghrelin, secreted mainly by cells in the stomach fundus, the upper part. Ghrelin levels rise before meals, stimulating appetite. It tells your brain, time to eat. It also has a role in stimulating growth hormone release, interestingly.
The hunger hormone. Okay, once food arrives in the stomach.
Then gastrin steps up. It's released by G cells in the stomach antrum, the lower part. Its main job is to stimulate the parietal cells to secrete gastric acid, HCl, and also chief cells to secrete pepsinogen, the precursor to the protein-digesting enzyme pepsin.
Acid and enzyme release. Got it.
But gastrin also has a trophic effect. It actually helps maintain the health and growth of the gastric mucosa itself. It keeps the lining strong.
Okay, so food gets acidified, churned into chyme, and starts moving into the duodenum. What happens then?
As soon as that acidic chyme, like pH below 3, hits the duodenum, it triggers the release of secretin. Secretin's job is basically damage control for acid.
How so?
It tells the stomach, whoa, slow down the acid production. And critically, it stimulates the pancreas to release a large volume of fluid that's rich in bicarbonate. Its main goal is to neutralize that incoming stomach acid, protecting the duodenal lining.
Makes sense. Neutralize the acid. What if fats and proteins arrive?
Ah, that's the cue for CCK, or cholecystokinin. Released from cells in the duodenum and jejunum when they detect protein and fat digestion products. CCK is a multitasking hormone.
What does it do?
3 main things. 1, it tells the gallbladder, contract, release bile. Bile is essential for fat digestion and absorption. 2, it stimulates the pancreas to release its digestive enzymes, lipase for fat, proteases for protein, amylase for carbs.
Okay, bile and enzymes. What's the third?
Satiety. CCK acts on the brain to help signal fullness, to reduce food intake. It tells you, okay, we've got nutrients coming in, you can probably stop eating now. It's a key player in appetite regulation.
Interesting. Any other major hormonal players we should mention?
Definitely the incretin hormones, primarily GLP-1, glucagon-like peptide 1, and GIP, glucose-dependent insulin-tropic polypeptide. These are released from the small intestine, especially after a meal rich in carbohydrates.
Incretins. What's their role?
Their main claim to fame is augmenting insulin release from the pancreas, but only when blood glucose is elevated. It's a glucose-dependent effect. This makes the insulin response to a meal much more effective. They also slow down gastric emptying a bit, helping to manage the rate of glucose absorption.
So they're really important for blood sugar control after eating.
Hugely important. And drugs mimicking GLP-1 action are now major therapies for type 2 diabetes and even weight loss.
Wow. Okay, so this whole system, nerves, hormones, is coordinating a massive amount of secretion, too.
Yeah.
How much fluid are we talking about daily?
The numbers are pretty staggering. Porth's notes estimate around 7,000 milliliters. That's 7 liters of fluid secreted into the GI tract each day. Saliva, gastric juice, pancreatic juice, bile, intestinal secretions. It adds up.
7 liters. But obviously we don't lose that much.
Not even close. Normally only about 100 to 200 milliliters of fluid is lost in the stool. The vast majority, over 95%, is efficiently reabsorbed, mostly in the small and large intestine. The body is incredibly good at recycling that fluid.
Which highlights why dehydration happens so fast if absorption fails, like in severe diarrhea. You're losing liters.
Precisely. Now, a key part of that secretion is the stomach acid. We mentioned gastrin stimulates it. It's made by parietal cells.
And they use that famous pump.
The H plus K plus MASH ATPase, the proton pump. It actively pumps hydrogen ions, protons, into the stomach lumen, making it incredibly acidic down to pH 1 or 2. Parietal cells also make intrinsic factor.
Intrinsic factor. What's that for again?
Absolutely essential for absorbing vitamin B12 further down in the ileum. Without intrinsic factor, you can't absorb B12, leading to pernicious anemia.
Okay, so parietal cells equals acid plus intrinsic factor. But with all that acid, how does the stomach not just digest itself?
Excellent question. It relies on the gastric mucosal barrier. This is a multi-layered defense. There's a thick layer of mucus, rich in bicarbonate, trapped within it, acting as a physical and chemical buffer.
Mucus and bicarb. Anything else?
Yes, the integrity of the cell membranes themselves, tight junctions between cells preventing acid leakage, and crucially, locally produced chemicals called prostaglandins.
Prostaglandins. Aren't those involved in inflammation and pain?
They are, but in the stomach, they are protective. They enhance mucus and bicarbonate secretion, they increase mucosal blood flow, which helps whisk away any acid that leaks through, and they promote cell repair. They're vital for maintaining that barrier.
Ah, okay. And this connects to common medications.
Directly. Things like aspirin and other NSAIDs, non-steroidal anti-inflammatory drugs, work by inhibiting cyclooxygenase, COX enzymes. These enzymes are needed to make prostaglandins.
So taking NSAIDs reduces the protective prostaglandins in the stomach lining.
Exactly. You take away that protection. Then the stomach acid can damage the underlying cells much more easily. Hydrogen ions get trapped inside the cells, causing injury, inflammation, necrosis, leading potentially to gastritis or peptic ulcers. It's a major side effect because you're undermining a fundamental defense mechanism.
That's a really clear link. Okay, let's shift focus downstream to the small intestine again. We talked about absorption being key there. Let's clarify digestion versus absorption.
Good distinction. Digestion is the process of breaking down large food molecules into smaller ones that can be absorbed. It's mainly done by enzymes through hydrolysis, using water to break chemical bonds.
Dismantling the food.
Right. Then absorption is the process of moving those small digested molecules across the mucosal lining, out of the lumen, and into the blood or the lymph fluid.
Moving the pieces into the body.
Exactly. And the small intestine is built for absorption. We mentioned the huge surface area, maybe 250 square meters, thanks to folds, those finger-like villi, and even microvilli on the cells themselves.
Massive area. And there are enzymes right there, too.
Yes, the enterocytes, the absorptive cells on the villi, secrete brush border enzymes. These enzymes actually stick to the surface of the microvilli, the brush border.
Why is that location important?
It means the very final step of digestion happens right at the site where absorption occurs. Super efficient. Disaccharides are broken into monosaccharides right where the monosaccharide transporters are. Small peptides are broken into amino acids right where the amino acid transporters are.
Smart design. Okay, let's compare how the major nutrients get absorbed. Carbohydrates first.
Carbs need to be broken down to monosaccharides, glucose, fructose, galactose to be absorbed. Fructose uses a facilitated transporter, but glucose and galactose, they rely on a specific transporter called SGLT1.
SGLT1, sodium glucose linked transporter 1.
That's it. And the sodium part is critical. SGLT1 uses the energy stored in the sodium gradient across the cell membrane, which is maintained by the sodium-potassium pump, to pull glucose and sodium into the cell together. It's a co-transporter.
So glucose absorption depends on sodium.
Absolutely. And water follows solutes. So when sodium and glucose are absorbed, water follows them osmotically.
And that's the principle behind oral rehydration solutions for diarrhea.
Precisely. You include glucose and sodium in the drink. The SGLT1 transporter actively pulls them into the intestinal cells, and water gets pulled along for the ride, helping to rehydrate the person even when their bowel function is disrupted. It leverages that specific transporter.
That's a fantastic clinical tie-in. Okay, what about fats? They seem more complicated.
They are. Large dietary fats, mostly triglycerides, are insoluble in water. First step is emulsification. Bile salts from the liver coat the fat globules, breaking them into smaller droplets. Stomach turning helps too. This increases the surface area for enzymes.
So, bile acts like detergent.
Good analogy. Then, pancreatic lipase, the main fat-digesting enzyme, can attack the triglycerides on the surface of these droplets, breaking them down into fatty acids and monoglycerides.
Okay, broken down, how do they get across the membrane? Still fatty.
Here's where bile salts play another role. They combine with the fatty acids and monoglycerides to form tiny aggregates called micelles. These are water-soluble on the outside, but carry the fatty product inside.
Like little delivery packages.
Exactly. Micelles diffuse through the unstirred water layer right up to the brush border membrane. The fatty acids and monoglycerides then diffuse out of the micelle and into the enterocyte.
Okay, they're inside the cell, now what?
Inside the cell, they get reassembled back into triglycerides. Then, these triglycerides, along with cholesterol and proteins, are packaged into much larger particles called chylomicrons.
Chylomicrons, and where do they go?
They're too big to enter the blood capillaries directly. Instead, they are extruded from the cell and enter the lymphatic capillaries within the villus called lacteals. The lymph fluid, now milky with these chylomicrons, eventually drains into the bloodstream. So fat absorption primarily goes via the lymph, not directly into the portal blood like carbs and proteins.
A completely different route. Fascinating. Now, we can't talk about the intestine, especially the lower part, without talking about the bacteria. The intestinal flora.
Or microbiome, as we often call it now. It's a vast and complex ecosystem, trillions of bacteria, vastly outnumbering our own cells, and mostly anaerobes, bacteria that live without oxygen, dominate in the colon.
And they're not just passively sitting there, right? They're active.
Incredibly active. They perform vital metabolic activities. They ferment dietary fiber and undigested carbohydrates that reach the colon, producing short-chain fatty acids which the colon cells actually use for energy. They synthesize certain vitamins, most notably vitamin K, which we need for blood clotting.
So they're helping us extract value and make nutrients.
Yes, and they have trophic effects. They help maintain the health and stimulate the turnover of the intestinal epithelial cells. They're also crucial for training our immune system.
And protection.
Huge role in colonization resistance. A healthy, established gut flora occupies the available niches and produces substances that inhibit the growth of potential pathogens. They basically make it hard for bad bacteria to get a foothold.
Which explains why broad-spectrum antibiotics can be a problem.
Exactly. If you wipe out large swaths of your normal protective flora with antibiotics, you open the door for opportunistic pathogens like Clostridium difficile, C. diff, to overgrow and cause serious infection and diarrhea. Disrupting that balance has consequences.
Okay, that brings us full circle back to the GI tract as an immune organ. You mentioned the barrier earlier, but there's specific immune tissue too.
Yes, a massive amount. Given that huge surface area, Porth's estimate is about 32 square meters, half a badminton court facing the outside world via the lumen. The immune surveillance has to be extensive.
32 square meters.
It's huge. The main system is called Gut-Associated Lymphoid Tissue. This includes organized structures like Peyer's patches, which are prominent clusters of lymphoid follicles found mainly in the ileum. They're packed with B cells and T cells ready to respond.
So like lymph nodes, but embedded in the gut wall.
Kind of, yeah. But they need a way to know what's in the lumen without letting pathogens invade. That's where specialized cells called M cells come in.
M cells, microfold cells.
Right. These are unique epithelial cells found overlying the Peyer's patches and other lymphoid follicles. They actively sample antigens, bits of bacteria, proteins directly from the lumen via endocytosis.
They reach out and grab stuff.
Essentially, yes. Then they transport these antigens across the cell and deliver them to antigen-presenting cells, APCs, like dendritic cells and macrophages, waiting just underneath. These APCs then process the antigen and present it to T cells, initiating an adaptive immune response if necessary. It's a controlled sampling mechanism.
Very clever. Any other key immune cells right at the front line?
Absolutely. There are also intraepithelial lymphocytes, or ILs. These are mostly T cells, but they actually live within the epithelial layer itself, scattered between the enterocytes.
Right in the barrier.
Right there. And the source highlights something important. Many of these ILs are unique. They don't necessarily need the typical priming process in lymphoid organs. They seem poised to react immediately upon encountering certain stress signals or antigens on infected epithelial cells. They can quickly release cytokines or kill infected cells. They're like rapid responders embedded in the wall.
Wow. So a multi-layered immune defense, physical barrier, chemical barrier, innate cells like IELs, and the organized adaptive system of GULT sampling via M cells.
You've got it. It's an incredibly sophisticated system protecting that vast interface.
Okay, so just to recap quickly, we've covered the fundamental structure, those 4 layers, plus a protective omentum. We looked at motility, the intrinsic slow wave setting the pace, the local control by the gut-brain, or ENS, and the modulation by the ANS.
Right, and the crucial chemical signaling. The GI tract as this huge endocrine organ, using hormones like ghrelin for hunger, gastrin for acid and growth, secretin for neutralization, CCK for digestion and satiety, and the incretins for insulin support.
We touched on the massive fluid secretion and reabsorption, the critical role of the gastric mucosal barrier, especially regarding NSAIDs, and the different mechanisms for absorbing carbs versus fats.
And wrapped up with the vital importance of the gut flora and the complex, multi-layered immune defenses patrolling that enormous surface area.
It really paints a picture of a system that's far more complex and active than just a simple tube.
Absolutely. It's constantly sensing, responding, adapting.
So for a final thought, something to leave our listeners mulling over. You mentioned CCK earlier, triggered by fats, slowing stomach emptying.
Yes, that response to fat and protein. It signals the gallbladder and pancreas, but also puts the brakes on the stomach.
How might that connect clinically? Thinking about something common, like reflux.
Okay, good connection. Think about someone prone to GERD, gastroesophageal reflux disease. They eat a meal high in fat.
Okay.
That high-fat content triggers a significant release of CCK. CCK, as we said, slows gastric emptying quite effectively, so the fatty meal stays in the stomach longer.
More volume, more time.
Exactly. More volume sitting there for longer builds up pressure within the stomach, and that increased intragastric pressure physically pushes against the lower esophageal sphincter, that valve at the top.
Making it more likely to open inappropriately and let acid splash back up.
Precisely. So the body's normal physiological response to dietary fat-releasing CCK to slow emptying can directly worsen reflux symptoms in someone who's susceptible, purely through that mechanical pressure effect. It's a direct link between the hormone, the motility change, and the clinical symptom.
Wow. It just shows how interconnected it all is. A hormone regulating digestion downstream directly impacts mechanics upstream.
It really does. The physiology underpins so much of the pathology.
That's a fantastic insight to end on. Thank you for walking us through that complex physiology today.
Pleasure.
And thank you, our listeners, for joining us on this deep dive into the GI system's foundations. We hope this exploration helps solidify your understanding. Keep exploring, keep questioning. The human body is endlessly fascinating.